Okay, so I'm going to tell you today a little bit about the Advanced BioImaging Facility, and then at the end we should have some time for questions and answers. If you have any questions about services we offer, services we may not offer but you think would be useful, or any clarification on anything that I'm presenting. Okay, so I wanted to start out with the people. So as you know, any advanced microscope isn't very helpful if you don't have the experts who know how to use it and train people how to use it and support them during their research. So right now we have Phil Kesner, who's an imaging scientist in the facility. He's been with us for about 1 year, and he did his PhD work at the MNI with the Ruth Heiser lab. We also have Joel Ryan, who's an imaging scientist and an image analyst, and he's been with us for 2 years. He actually was a lab manager and worked for a while with Paul Maddox, who many of you may know from University of Montreal, and then he did his PhD in Germany. We have Barbara Doroka, who I see is attending here today. She's an imaging scientist as well, and she's been focusing on training and education initiatives, and she's the one who developed our fully automated step-by-step remote training on the AxioVert 3 that many of you may have attended. Then there's myself, who basically tries to keep everything running and keep us funded. Anne-Marie Ladouceur and Nellie Villeman are both imaging scientists. Anne-Marie is our Assistant Director of Training and Education, and Nellie is an image analyst, and they're both currently on leave. So if you work with the ABIF, we have a little motto that we actually, we should use more than we do. It's from planning to publishing and everything in between. And so we really encourage people to come to us early. We have free one-on-one consultations where we talk about experimental planning, sample preparation, training on the microscopes. We work with the users to find the best microscope for their application. And then we do additional consultations and training as needed. So someone, for instance, may want to train on the confocal, but then a month later they want to move to live samples. And so they would have a second training on how to image live samples on the confocal. We also have image analysis, so developing image analysis strategies using some of the advanced software we have available at the facility. And we also offer custom image analysis algorithm development. For publishing, we're happy to help make figures, interpret data, or plan additional experiments, say you have comments from reviewers or a grant that you want to address. And we really try to offer ongoing support through the entire project. I will mention we have a YouTube channel now that has several videos on it. We have some training in ImageJ Fiji and some other training videos there. And we also have the ABIF Twitter site where we try to tweet out interesting things that may be happening in the facility as well as in other communities on scientific Twitter. So what do we offer? We offer training. So our major, I'll say a little bit more later, but our major training is one-on-one. And we do about 250 of those a year. We basically train people how to use the microscopes and do image analysis. And then I'll say a little bit more about courses and workshops after. They've been mostly virtual since the pandemic, but we have plans to move back to more in-person events in 2023. For technologies, I'll give you an overview of what we have, but as a snapshot, we have wide field, confocal, we have a lot of automated microscopes, and actually intravital imaging is something we're not offering anymore. My apologies, I should have updated that slide. The applications we have are diverse, but just to mention a few, we have live cell imaging, biophysical tools, measuring dynamic interactions, and statistics and big data is an area we've been getting into more recently. So if we just look at the numbers for 2021, as I mentioned, we have about 250 trainings per year. We have 230 unique users from 76 labs, 21 departments, and 4 faculties. We currently have 12 microscopes, 5 advanced image analysis workstations, and we have a 200 terabyte file server for image storage. We have about 30 publications per year, about 12,000 hours of use, and about 5,000 imaging sessions per year. So that works out to about 14 imaging sessions every day. So a little more detail on the instruments we have. We have wide field, which includes bright field and fluorescence imaging, confocal laser scanning microscopy, and spinning disc confocal microscopy. We have high content imaging where it's fully automated and you can use multi-well plates. And this includes both wide field imaging and spinning disc. We have fluorescence lifetime imaging. And in the fall, we should be receiving a lattice light sheet microscope, which I'm sure we'll do an Illuminate session once we have that in place and installed for people to see what it's about. But it's really an instrument for high-speed, high-resolution imaging of cultured cells, spheroids, tissues, going 50 to 100 microns into the sample. So it's not meant for very large samples, like a whole mouse brain and things like that. It's for higher resolution, but with smaller samples. And it's very gentle for live imaging, so there's very little phototoxicity. And I'll just mention that the Lightsheet Z1 microscope from Zeiss is no longer under our management. So if anyone wants information about that instrument, I can talk to them offline, or you can email me. So some of the other applications we have that are less mainstream are polarization microscopy. I'll show you an example of that. Differential interference contrast, phase contrast, and dark field. I'll show you some nice examples of spectral imaging, which can be used for multicolor imaging as well as for fret-based probes. Total internal reflection fluorescence microscopy, which is designed for looking at things that are very close to the cover slip. And then on the software side, we have Metamorph, Amaris, deconvolution algorithms, and we also have a lot of support for ImageJ Fiji if people like using that open source tool. So I've already mentioned some of these, but some of the applications, I think one of our real specialties is live cell imaging. We have a lot of experience there in how to optimize things to keep your cells happy and keep phototoxicity at a minimum. Another slide I needed to update, my apologies. The SIM super resolution is no longer in the facility either. That instrument was decommissioned, and if anybody's interested in that technology, I can also point you to where you can find that around McGill. Fluorescence resonance energy transfer FLIM, 3D morphology time-lapse, the high-speed live imaging I mentioned, the lattice light sheet will be coming soon. I'll show you a nice example of 7-color imaging, and we can do rapid 3D imaging of large samples, the 3D imaging of entire small organisms. High-resolution multicolor imaging is also possible. And we have a motorized stage on most of our instruments, so you can do multi-point time-lapse, tiling of large samples. And another aspect I think we really specialize in is the quantitative imaging, so really helping people collect quantitative data and then analyze that data in a way that it's quantitative and maintains the integrity of the data. So for training and education, as I mentioned, we have the consultations. We also have the fully remote step-by-step wide field training, and we have plans to expand that training over the summer to our confocal laser microscopes. We have these Virtual Illuminate seminars. Again, I would hope we'll go back to in-person Illuminate seminars in the fall. A nice project that we have in collaboration with the imaging facility at the Neuro is an Amiro National Database, and we're working on setting up data storage in the cloud, which will include all of the information about the images and the image processing so that the data can be easily shared and reused. And if anybody's interested in that aspect or has data they want to put in the cloud and share with collaborators or just with the general community, please reach out to me, as we're piloting some data sets right now. And once we have that ready, we'll certainly be letting our user base know. And there is a nice a couple of Amiro training videos on the ABIF YouTube channel. The Montreal Light Microscopy courses should come back in 2023 as well. We'll likely do our introductory fundamentals course over the March break in 2023. And then we'll probably look to do a course on the lattice light sheet, since that'll be a new instrument to the facility. And the last point I wanted to make here is, over the pandemic, we've gotten quite involved with national and international groups working on research data management. And Joel from the facility has developed a tool called Methods J 2. And what it is it will collect the metadata from the images about how the image was collected. And it will collect metadata from a hardware file that we can provide to the users. And it will write your method section for a manuscript. So you'll get a first draft of your method section. Right now it's available for our AxioVert 3 microscope, but we're working on getting the data together for all of our microscopes. And we have a comprehensive research data management workshop that we did as part of the Images to Knowledge meeting in Wisconsin that was just held in May. And that'll be becoming available as a tutorial, a virtual tutorial that people can take on their own, or can reach out to us if they want to work with us on that. So I'll be happy to send more information once that information is packaged. So it's got sample videos, descriptions of how to use the tools, and sample videos of all the steps. So it's really easy to follow and work with. And I've included the link there to the Methods J2 GitHub, where you can also read lots more about this application. And it's just a plugin for Fiji, so you can download it and run it in Fiji. So the last sort of aspect of education and training is I was fortunate to be part of this quantitative confocal article, which came out last year, and it was really fun to write with 4 other imaging facility directors from around the world. And I would really highly recommend people take a look at it. It has a lot of great tips and advice. And I just show one example here. This is a figure from Allison North at the Rockefeller University. And she's looking at the same sample prepared the same way, but with different mounting media. So you can really see the differences here. She has a membrane marker here in 405, microtubules in 488, and the actin cytoskeleton in 647. And you can see in VectaShield that the 2 cytoskeleton dyes are totally quenched. You can see this batch of glycerol has been contaminated, or there's some artifact in it that it's labeling the nuclei, whereas when she prepared new samples with a new batch of glycerol, the nuclear staining disappeared. So this one was really interesting to me, having been in the field for a long time. I think if I saw the second panel there, I would have thought I did something wrong with my staining, and I would probably go back and tweak the staining parameters. So I think this is a really great example of the things you need to look for if you're troubleshooting sample preparation. So I'm going to move now to just highlight some selected projects from the facility. I will give a disclaimer up front that a lot of them are from my lab or my collaborations with different labs in the life sciences complex. We'd really love to build up this slide deck with lots of examples of people using the facility. So if anybody be interested in providing a couple slides of data that we can add to the selected projects, I'd be really happy to work with you on that. So it's not comprehensive. It's not going to show you everything we have, but I think it'll be a nice snapshot. So the first example is polarization microscopy. This sample was prepared in the Park lab, and it's a breast cancer tissue slice, and they labeled it with a dye called Picocyrus red. And when you look at it with polarization microscopy, the different types of collagen show up in different colors. So the thicker collagen fibers are showing up here in this orangish red, and the thinner collagen fibers are showing up in green. And this is just done on one of our standard wide field microscopes using polarizers. Another straightforward service or application we offer is wide field deconvolution. And this is very underutilized, I think. If anybody's doing imaging of relatively thin samples, 10 to 20 microns, then wide field deconvolution is a great option. And you can see this is just an example of cultured cells, which are labeled with actin in green, mitochondria in red, and the nucleus in blue. And you can see when we do the deconvolution, what this does is it reassigns the out-of-focus light to the different structures. And you can see how we have much, much better contrast, much better signal to noise. And if we now want to do 3D rendering of those structures, it's much easier to do on the deconvolved data. This is another really nice example from the Park Lab. This is a 7-color staining of breast cancer tissue. And again, this is a tissue slice on a microscope slide. You can see many immune markers. They also have DAPI, which is not shown on this image, but you can see these 6 colors very well separated and nice signal to noise for each of them. And then this is the overlay of those 6 colors. Again, the DAPI is not shown here. So you can really see the different structures and the different cell types in the tissue. And this is done using spectral imaging and unmixing of the signals. As I mentioned before, live imaging is one of our specialties. So this is an example of live cell wide field imaging. So just looking at them on one of the axiovert microscopes. And you can see the cells moving, growing, dividing. On the left side are the control cells, and on the right side we've put in a phosphomutant, and it really slows down the cells when this protein can't be phosphorylated, and they move a lot slower. So on the next slide, I'll show you the quantitative analysis of these videos. So we do tracking of each individual cell using Metamorph software. And then each cell is assigned a position of 0, 0 from the first frame in the movie, and then the movement is measured relative to that start position. And you can see that the wild type control cells are moving much, much faster and much further than the cells expressing the phosphomutant. We also have high content imaging, so the ability to image on multi-well plates. This is an example using a 24-well plate, and this is a spitting disc confocal microscope, so it actually has 3D sectioning capabilities as well. We also have a wide field system in the facility that can do up to 5-color imaging. This one is a spitting disc, so you have that extra sectioning ability, and it's going to be a little bit better. Well, I guess depending how you set up, it could be good for live cell imaging as well. And this was an example with a student in Peter Siegel's lab, which I was co-supervising, and they imaged these cells on the microscope. And this is actually a special feature on the PerkinElmer microscope called digital phase contrast. And you can see the image on the left and the image on the right. The one on the right is this digital phase contrast. So there's no fluorescent marker here. It's just a natural or an optical way of creating contrast that is white on a dark background. And then they have a nice algorithm for tracking the cells automatically. So this is a video using this digital contrast, and then you can see each cell is being tracked throughout the video. And on the right you can see control and treated cells, where when the cells are treated with growth factor, they move much faster. And you can see every dot is a single cell that's been tracked. The other expertise we have with live cell imaging is measuring phototoxicity. So if anybody is doing live imaging and you want to make sure that your protocols are not causing a lot of damage to your samples, we have a few different assays that we can help you with. This one is showing mitochondrial staining. It's a dye called MitoTracker Red. And you can see in the top panel we have low light levels on the sample, and over 4 hours the mitochondria are still looking very healthy, and they form these elongated fibers and smaller structures. Whereas when we put high light intensity on them, even after 45 minutes to an hour, you can see the mitochondria are starting to condense and basically lose their structure, and this is a sign of oxidative stress in the cells. This is the quantification of this kind of data. So this is just looking at the area of the mitochondria over time, and you can see that they get much smaller and round up into these structures with phototoxicity. Another way to measure phototoxicity is directly measuring reactive oxygen species. So on the left side, we take an image of this Cell Rocks deep red dye before we do our imaging. And then on the right side, we've taken 400 images at relatively high intensity. And you can see the dye is lighting up now. And it does also localize to the mitochondria. So you can see there's been a lot of damage done here. And you can see that this dye is very quantitative in the sense that the higher power, the more light you put in on the sample, the more reactive oxygen species are produced, and the higher intensity the dye. I'll also show you a few examples of adhesion dynamics. So this is one of the key areas we use in my lab. These are spinning disk. This is a spinning disk movie of Paxil and GFP, which localizes to adhesions in cells. And we're interested in measuring the characteristics and dynamics of those structures to understand the regulation of cell migration. So this is another example here showing quantitative imaging as well. So this is now total internal reflection fluorescence. So we're only looking at about 100 nanometers from the cover slip. And then we're zooming in on this protrusive area of the cell here. And we use a MARIS software to isolate and identify the adhesions, track them, and measure their intensity. We then can take that intensity information into Excel. And my student Alex wrote a custom code, which is available for MATLAB, where it identifies periods where the adhesion is disassembling, periods where it's stable, and periods where it's assembling. And then using a semi-log plot of the data in these disassembly and assembly periods, we can calculate the rate of adhesion formation or adhesion disassembly. And this is an example from his project where we were looking at the assembly rates in green and the disassembly rates in red. And we were looking at the effect of growth factor treatment. So similar to the other example I showed you, when the cells are treated with growth factor, they move faster. Well, the adhesions are also much more dynamic, so the rate of assembly and disassembly is much higher. And then the protein he was interested in his project is lipoma preferred partner, or LPP. And you can see when the LPP is knocked out or knocked down in the cells, the adhesion dynamics do not respond to the growth factor. So you see the adhesions are still somewhat dynamic, but they're unable to respond to the treatment. And you can also see from this plot, these are box plots, but showing the individual data points from every adhesion, that we have thousands of data points. So this is something we can also work with people if they want to really characterize these distributions and understand what might be happening in more depth than just looking at individual means or averages. We can help with that. So along that same line, this was another project with Rebecca Deagle and Luke McCaffrey's lab at the Cancer Center, where we were looking at adhesions on 3D substrates with different characteristics. And she was looking at the adhesion characteristics in a spatial way. So the area, the distance from the edge of the cell, how circular they were, how solid they are, and their length. So solidity is an interesting term. It's, if you look at this shape here, this adhesion is not very solid because when you make a convex hull around it, there's a lot of area that doesn't include adhesions. Whereas this adhesion here is very solid. It doesn't have much black area in this surface. So it's a measure of sort of how rough the edges of the adhesion are, how bent it might be, how irregular the structure might be. And what Rebecca did is she combined data from many different conditions and many different cells and used hierarchical cluster analysis in order to see if the data would cluster into different populations in a similar way that many people use for genomic or proteomic data. So these cells were grown on a 3D surface of bovine skin collagen. They were a breast cancer epithelial cell expressing the LPP protein I already mentioned. And then she used these binary maps of adhesions to measure all these different adhesion characteristics. So another tool that she used, which everybody at McGill has access to, is a software called JMP. And this software is linked with Excel. So if you have data in Excel, you can link it to JMP and make these kind of plots. And what she did here is she broke down the data into the different classes. So if you see here, there's A, B, C, D, 4 primary clusters, but then there's 9 subclusters. And so this is that same data broken down into the 9 subclusters. And these plots are really fantastic. You can put in a huge amount of information. So I'll just walk through it relatively slowly just so it's clear. So the size of the spot itself is the size of the adhesion. So the larger the spot, the larger relative size of the adhesion. The color coding is the distance from the cell edge. So the blue spots are close to the cell edge, and the yellow to red, I guess yellow are sort of halfway between the middle of the cell and the edge, and the red spots are really in the middle of the cell. And you can see quite a bit of difference in the colors as you look across the different clusters. And then the y-axis here is the circularity, so how round are the adhesions. And so we have some schematics here kind of showing what the different types of adhesions would look like for the different clusters. And I won't go through all of them, but just highlight a couple. So if we look here on this end, we see that the adhesions are quite long. They're not very circular. They're elongated and they're really large. So they might look something like this in the cartoon. If we go all the way to the other end, the adhesions are quite small, and these are mostly bluish in color, so they're close to the edge of the cell. And then if we look at cluster 6, kind of in the middle, we can see that we see much more widely distributed adhesions across the cell, but they're also still fairly small. And so it would be something more like this, where you have adhesions everywhere, but they're still relatively small. So you can see that you can get a tremendous amount of information from these kinds of higher order data analysis. And this is the same a data set from the same set of experiments, but now the LPP protein has been mutated and it can no longer bind to the actin cytoskeleton. And there's several phenotypes you can kind of see by comparing the two data sets, but the most obvious is that we've lost the cluster number 1 completely. So there's no cells with these big long adhesions, which is fitting if you've disrupted the interaction with the cytoskeleton. So again, you can see how with this kind of data, you can really explore the data in more depth and see subtle phenotypes. So spectral imaging, I showed you already the 7-color imaging example from the Park lab in breast cancer. You can also use it to get rid of autofluorescence. So this is an example of C. elegans, where they have a GFP protein expressed, but there's a lot of autofluorescence in the worm. So you can see here, this is a zoomed out view, and then if we zoom in here, you see a lot of green. But we can do spectral fingerprinting, so we can image a sample that has just GFP and image a sample that has just autofluorescence, and this is the intensity of signal over wavelength, or the spectra. And then we can unmix that mathematically, and what you see now is the GFP is only in these bright spots and at the surface, whereas the autofluorescence is kind of all over. And you have the overlay here, and I'll apologize, I shouldn't have used red and green there for anybody who might be colorblind, but you can see the difference here in the individual panels. Another application we have for spectral imaging is FRET, or fluorescence resonance energy transfer. This is an example of a biosensor where you have GFP and RFP, and they're linked together by a little piece of spider silk protein, which is an elastic protein. And they're in the middle of the talin protein. So talin is a protein that links adhesions to the actin cytoskeleton. And if the protein is relaxed and not under tension, then the spring will be relaxed and you get FRET between GFP and RFP. And if the protein is under tension, then you will have them separate and you'll have reduced FRET as the molecules move further apart. The examples shown here are liver-derived cancer cells from mouse. And in the top panel, this cell is from sample that's not treated with growth factor. And in the bottom panel, you have cells that are treated with growth factor. And you can see how the intensity is much higher, which is corresponding to lower FRET and higher tension after the growth factor treatment. And then you can see, if we zoom into this area of the cell that's protrusive, you can really see the difference in the amount of tension on the adhesions. You also will probably see that in the growth factor treated cells, the adhesions are also more numerous and a bit smaller than in the control sample. And then this plot here is a violin plot, which shows the distributions of all the data points from many different cells in the experiment. And what you see here is this shift in the distribution to lower fret when the TGF-beta treatment happens. And that lower fret is corresponding to higher tension. So this sort of peak here would be corresponding to a cell like this. We also had a couple of other experiments. If the LPP protein is knocked down, you don't see this decrease in fret or increase in tension. And then we have a control sample, which is basically the talon that's just with the end terminal linking to the adhesion, and this part of the talon is missing, so it can't interact with the cytoskeleton. So it's always in this low tension, high fret format, and you can see the fret ratios stay high regardless of the growth factor treatment. So I'll apologize in advance if anybody gets motion sickness from Amaris. I know it, it gives me motion sickness sometimes. This is an example from the Masoner lab in the McIntyre building in the pharmacology department. And they were interested in lipid droplets and lysosomes. And they had a paper that was, they were trying to publish, and the reviewers were concerned that the lipid droplets weren't actually inside the lysosomes. But when we did the 3D rendering and looked at it in Amaris, we could very clearly show some of the lipid droplets were completely engulfed inside the lysosomes. So this really helped them to address their reviewers' concerns quite quickly and then develop a nice visual and some figures for the paper. This is another example from Amaris 3D imaging. This is a sample from the McCaffrey lab using the laser scanning confocal. It's a 3D stack of epithelial cell spheroid, and the cell mask is labeling the membranes in green. Mitotracker red is in magenta, and a nuclear dye in blue. And in the first panel, you see the whole spheroid. It's about 30 micrometers from the surface is the cut through that's shown. And in B, you're seeing the surface rendering in Amaris of the three different stains. In C, it's turned just a little bit so you can start to see some of these surface details of the membrane. And then in D, we're just zooming right in and just showing the membrane. So you can see the membrane between cells, and then again, you can see these details on the surface of the spheroid. I just wanted to mention briefly, we have been getting a little bit into AI machine learning. There's a lot of really nice noise filters that are available now to remove noise from images while keeping the real signal from your sample. And this is a project in collaboration with the Lavoie Cardinal lab at Laval University, where we wanted to work on generating synthetic images. So the idea here was to take images of adhesions and generate synthetic images of the actin cytoskeleton. So the actin cytoskeleton is something that's really representative of how mobile a cell will be. So in the top panel, we have a paxillin mutant that cannot be phosphorylated, and the cells are not very motile. They form these really big actin cables, and they're very stable. They don't move very much. Whereas when we put in a phosphomimic in the cells have a much less organized actin cytoskeleton, and they're much more migratory. So we were thinking just looking at the actin cytoskeleton, you could predict how migratory a cell might be. So what we did here is we had cells that were expressing paxillin EGFP, and we stained them with actin, phalloidin 405. And we particularly used the 405 because it's a blue dye and gives us much better resolution on the microscope than red or far red dyes. And then this is the overlay of the two signals. And we used what's called an adversarial network to train the network that if the adhesions look like this, the actin looks like this. And then after you do the training, you're able to feed the algorithm an image, and it can generate a simulated actin image. And this is basically what the simulated or synthetic actin image looks like for that cell. And then you can compare that synthetic image to the real image, and you can see that it's actually doing a pretty good job guessing what the actin cytoskeleton might look like. So this is pretty preliminary data, but it's an interesting project, just applying AI machine learning to image analysis, and in this case, generation of synthetic images. And I believe this is my last slide. I just wanted to mention again the research data management and reproducibility. There's a really big international initiative now to address this problem, and I think everybody wants to do really high quality reproducible data. Everybody wants to share their images, but sharing image data is really complicated if you don't have the tools to annotate and curate and provide all the information that somebody else would need in order to reuse your images. So we were quite happy in December 2021, Nature Methods decided to publish a whole issue on reporting and reproducibility in microscopy. And we were on 6 of the 9 papers that were in the article, or in the issue. And one of them is QUARAP-LIMI, which is a quantitative microscopy international consortium, has about 300 or 400 members now from all over the world. And it's really focused on developing procedures, protocols, and methods so that people can quality control their microscopes, quality control their image analysis pipelines, sample prep, making figures, and really collect the best data possible, report the best results, and then be able to have the tools to share the data. I've also been involved in 2 other aspects of that issue and of this sort of topic. And one is a model that was published in order to really list all of the specifications you need to characterize an image. So kind of what are the minimum qualifications or minimum specifications you need in order for someone else to take that image and reuse it. And then I already mentioned a little bit this Methods J2 tool that Joel has developed for Fiji. And this is one of the articles in the issue where it gives a detailed step-by-step guide as to how to use that tool in order to generate methods text. And that's what I have for today. So I know I've been doing all the talking, so I'm very happy to take any questions and give any further information if people have things they're interested to hear more about. And I'm going to stop sharing and stop recording